Hello. Now we are on exercise 4 in your lab manual. This is part of your Diffusion Osmosis Lab. So this activity is focused on playing around with the process of osmosis. And you really want to keep in your mind that osmosis is defined as the movement only of water through a semi-permeable membrane. So to experiment with just osmosis, we're going to make a cell, a pretend cell. And we're going to make a cell out of this material called dialysis tubing. I have some here. It's in a beaker of water. I don't know how well you can see that stuff down here at the bottom, right? And if it's not in water, dialysis tubing looks like this. It kind of looks like cellophane tape. It's funky stuff, right? You might be thinking, well, how's that going to make a cell? Well, when you soak this stuff in water, it becomes very slippery and kind of squishy almost, right? So I pulled a length of it out here. And what's kind of fun is if you kind of carefully roll it between your fingers, you'll see you can make a little opening. So this truly is like a tube, so that you can, and at this point what I'm really doing is I'm on page 46 of your lab manual, right? It says obtain a short piece of dialysis tubing. That's what this is. Fold over the end. So you fold it once, you fold it twice, and then you clip it. So I have these funky little clips. I'm going to clip it right across those folds. And now I've essentially made a little, what was a tube and would have just, if I put any liquid in it, would have just come right out the other end. Now it should be stopped, right? Then it says the bag should be limp. Well, that looks pretty limp, right? That was number 4. And it also says on page 46, I'm looking down if you're wondering what you're looking at. Number 3, fill the bag with 10 milliliters of cell meal. Okay. The stuff in this little flask here is what is called cell meal, which if you read on page 46, it's just a mixture of water, glucose or sugar, egg albumin, which we talked about before in biomolecules, what kind of a biomolecule albumin is, starch, and then again water. And they even put a little question mark by the water because they want to know, like, if percent means per 100, then how much water percentage-wise do you have in this mixture? If you know it's 25% glucose, 0.5% egg albumin, 1% starch, then how much water? Hint, do some addition and then some subtraction, and you should be able to figure that out. Good thing to know. Maybe it shows up on your post-lab quiz, right? You're going to add 10 milliliters of this to the little cell baggy thing we've made. So how are we going to do that? Okay, I'm back with a different pipette that should not lose its liquid. Again, I'm going to draw up. Oh, I'm already at the top. Hold on a second. Even teacher sometimes has to experiment, right?
Teachers aren't perfect.
Nope. Okay, here we go. So here's the little opening. Now that might be hard to see. I got the pipette tip in now, and I'm pushing down sort of on the plunger at the same time I'm holding this off. So that there is 10 milliliters, of cell meal. And then it says clip the top of the bag. So I'm just going to fold it over again, and then I'm going to clip it. I'll clip you, just like this. It is a bit leaky on one side, but I think it's holding it. Okay, and then it says weigh the cell and record your results. So I have a scale here. Turn it on. And I'm going to make sure it goes to 0.00. Put my little cell thing there in the weigh boat. And it tells me right now my weight is 23.61 grams. So that's what you want to write down here, where it says what's the weight of your little cell. Okay? That's what you're going to do. Then it says, and now I'm on to step 7. You're going to soak that cell, this baggy thing, in this stuff called iodine solution. So I have a beaker. I have a bottle of this stuff called iodine solution. This truly is just a mixture of water and iodine. Pour some in. Ooh, isn't that a lovely, here I'll hold it up for you all to see, sort of dark urine color. Need more water, huh? So that's the color of the iodine. And what I'm going to do then is I'm going to drop this into here, and I'm going to videotape what happens to it. So just imagine right now I'm dropping it in, and then I'm going to videotape it. I'm going to make another setup, just so you know. I'm going to make another setup of the exact same thing. So I'm going to make another cell that I set up with the dialysis tubing, with the clips. But instead of soaking it into the iodine, I'm actually just going to soak it in distilled water. And the reason I'm making that tube will become clear to you later.